Hello friends, welcome to VET innovation classes. So in the previous class we are discussing about GPCR, G protein coupled receptors. Basically they are the super family of the proteins which consist 7 helical structure, and G means guanine nucleotide containing protein which have alpha, beta and gamma subunits, in which the alpha subunit is also attached with the GDP protein, guanine diphosphate, and after the conjunction of particular ligand, the alpha GDP complex will dissociate and GDP was transferred by GTP, and then the action will be happened. So in that we discuss about the cyclic AMP system, in which the adenyl G protein is stimulating production of cyclic AMP from the adenylate cyclase enzyme, which phosphorylating the different protein and providing cellular effects. Now second effector system is phospholipase C IP3 DAG system. Basically IP3 DAG are two different secondary neurotransmitters, secondary messengers, which are acting via this system. We can understand with help of this diagram. This is the our GPCR. GPCR means G protein coupled receptor, in which 3 subunits are attached: beta, gamma and alpha, and at the alpha side GDP is attached. And this is the extracellular membrane, this is the intracellular membrane, and as the adenylate cyclase enzyme present on the surface, same phospholipase protein also present on the surface of the cells. So PLC is indicating phospholipase protein, which, so when any drug or ligand bind with the receptor, then the G protein is activated, and G protein activated and stimulate the phospholipase C, which convert PIP2 into 2 secondary messenger, one is IP3 and second is DAG. Here PIP2 stand for phosphatidyl inositol biphosphate, and IP3 stand for inositol 1,3,5 phosphate, because 3 phosphate are attached with the inositol structure, and DAG here stand for diacyl glycerol. So friends, when the GPCR activated, it provide a stimulating effect on the PLC, phospholipase C protein, which convert PIP2 into 2 secondary messenger, one is DAG and second is IP3. So here IP3 work with the 2 mechanism. It basically release the calcium from the endoplasmic reticulum, smooth muscles of the endoplasmic reticulum where calcium channels are present. So it affect to the calcium channels, which increase in the calcium influx into the cells. So here the calcium come from the endoplasmic reticulum via calcium channels, which again act via 2 mechanisms. One from CAM means colmodulin channels, which are the channels of calcium, and colmodulin channels provide different type of cellular effects via contraction in smooth muscles, etc. And also calcium activate the phosphokinase C, as well as cyclic AMP, or also stimulating the phosphokinase C from changing inactive to active state. Same The calcium channels or increased concentration of calcium also activate the phosphokinase C, which can again phospholize the protein, and phosphorized protein are again produce the different type of cellular effects. So this is the IP3 secondary messenger system. Basically, after formation, it act on the calcium channel, release the calcium from the endoplasmic reticulum, and act via two channels, calmodulin channels, which are a specific channel for calcium, and provide cellular effects. And again in the second phase, it activate the phosphokinase C enzyme, which phosphorylate the different type of proteins and provide cellular effects. And same DAG is formed, and DAG also work on the phosphokinase C. It also change the state of phosphokinase from inactive to active stage and act via phosphorylation of the different type of protein. So friends, basically this is the IP3 DAG system. Now, what are the effects, pharmacological effects produced by the IP3 and DAG system? Via some of the effects produced by the IP3 system, like smooth muscles contractions via IP3 system, and also increase force of contraction of cardiac muscles, which can provide the positive inotropic effect on the cardiac muscles and increase the contractility of the cardiac muscles, and then cardiac output will increase. And also it is responsible for the secretion of exocrine glands and synthesize and release of the hormones also from endocrine glands. So basically it acts on both exocrine and endocrine glands, and also it is responsible for the release of neurotransmitters from the neurons. So IP3 involved in the different type of mechanism, means these actions were happened via G protein coupled receptor activating IP3 secondary messenger system. And also in case of DAG, it is also responsible for smooth muscles contraction, but not only contraction but also relaxation. Relaxation is mediated via DAG system, not IP3 system, and it also can increase or decrease in release of neurotransmitters from the neurons, because IP3 system is only working calcium also. So calcium cause contraction in the smooth muscles and different synaptic vesicles, which can release from the, because neurotransmitters are released in stored in the synaptic vesicles, and after contraction they can release from the outside synaptic vesicles. But DAG can provide the positive and negative effect both. So it can decrease or increase the release of neurotransmitter from the neurons, and also hormones from the endocrine glands, and it is also responsible for the inflammatory responses. So it is the important secondary messenger channel, which is also important for exam point of view. Now one more, this is the third system involved in the GPCR mechanism effector system. First, we are discussing about the enzyme linked effector system, in which the first is cyclic AMP, second is IP3 DAG system, and third one is phospholipase A2 or called AA system, in which what happened, again this is the GPCR, and here your PLC is present and PIP2. So one more surface protein PLC is also present another side, which can activate it via G protein. So after the activation of G protein, it can also formation of arachnoid acids. AA is stand for arachidonic acid from the phospholipase C protein, which is a surface protein present on the cell surface. So after the formation of AA, it can again converted to eicosanoids. Eicosanoids basically are three major particles are involved in eicosanoids. You all know, like PGs, prostaglandins, and thromboxane, and also leukotrienes. So these all factors are responsible for producing different type of cellular effects. So this is called arachnoid acid system or phospholipase A2 system, because in this the G protein is responsible for the formation of arachnoid acid, which again converted or metabolized into different type of eicosanoids like prostaglandins. We all know prostaglandins and thromboxane and leukotrienes have different type of cellular responses, like prostaglandins involved in the contraction of the smooth muscles and different type of gynec conditions, and thromboxane is responsible for the platelet activations in the red blood cells, and leukotrienes is also responsible for various type of inflammatory and allergic conditions. So basically these involve in the different type of cellular effects via AA system, and this system is again supported via PIP2 also, because PLC is converting PIP2 into two form, one is DAG and second is IP3, and IP3 and DAG both are acting on PKC, phosphokinase C, and phosphokinase C again responsible for the formation of arachnoids from the PLC. So it also affect the positive effects. So this is the AA system. So friend, this is the stimulatory effect of the G proteins, while the GS protein are involving in this mechanism, G stimulatory. But in some cases it also be inhibited via G inhibitory protein. If G protein inhibitory protein is involving, so this mechanism can be reversed or can be blocked via G inhibitory proteins. If G inhibitory protein is activating, so it can stop and decrease the production of these secondary messengers IP3 and DAG from the PIP2. Same in case of here also. If G inhibitory protein is involving, so it can also reduce the formation of arachnoid acid from the PLC. And now if we talk about what are the cellular effects produced by this system, so arachnoid acid itself a metabolites, arachnoid acid itself and its metabolites like PG, leukotrienes and thromboxane, a function as a intracellular messenger for controlling potassium channels in functions in certain type of neurons. Different type of neurons में ये potassium channels को operate करते हैं via arachnoid acid खुद भी करता है, और इसके जो producing metabolites है PG, thromboxane, leukotrienes, they all are working on the potassium channel and responsible for the different type of cellular mechanism. So guys, these all three type of effector systems are enzyme linked. This is the only first part of the G protein effector system, which is enzyme linked. Second type of action will produce via ion channel linked effector system. As we discussed in the ion channels are modulating via two ways. First is direct modulation, which are responsible for the different type of cellular effects, because the direct modulation is occurring via the different type of ion channels. Blockers are attacking on the surface protein present on the ion channel directly. So this is the combination of the direct receptor and drug is directly, but in the indirect action the ion channel are mediated via different type of receptors. They call ligand gated, and these are calling voltage gated. So in the ligand gated ion channel effector system, how we can understand? They are activating via G protein. Here again the structure of G protein, alpha, GDP. So GTP transfer from the GDP from the alpha binding site, and here as we know the ion channels are also present on the surface. But the stimulation for the occurring of the action is not depend on the ligand bind with the ion channels. The ligand will bind on the GPCRs, or drug will be bind on the GPCR. As the drug bind with the GPR receptor, again here the alpha and GDP, it will change into alpha and GTP, and this is responsible for the activation of these ion channels. When the ion channels open, it will open for the certain type of ions, and the ions will cross the membrane via ion channels, and when the GDP again catalyze into the GDP and the back to form GDP alpha complex. The ion channels will be remain closed for the certain type of ions. So this is the simple ion link effector system, in which the GS, it depend on the different type of proteins, the what type of protein is activating. If G stimulatory protein are activating, so that will responsible for the depolarization. Ion channels can produce two type of effect, as we know, depolarization, and like if sodium channels are involving, and hyperpolarization if they are involvement of chloride channels. So, and also it is selectively open for the calcium channels. So, depend on the what type of channels are activating and what type of ions are coming into the cells. So, G stimulatory open the calcium channels in myocardium and skeletal muscles. If the GS protein is involving, so that will open the channels of the for the calcium in myocardium and increase the contraction of the myocardium. So, there will be more cardiac output due to the high contraction in the heart. And if G naught or GI, G inhibitory or G naught protein are involving, so as G inhibitory produce the negative effect, so it will open the potassium channel, which also cause hyperpolarization in the heart and smooth muscles. So it will decrease the contraction of heart and the smooth muscles, as well as close calcium channel in the neuron. Means agar G naught and G inhibitory protein are involving, that will close the channel for the calcium, while GS is open the channels for the calcium. And also G naught and G inhibitory open the channels for calcium, potassium, sorry. So potassium will be hyperpolarize the cells, and sodium will be or calcium will be depolarize the cells. So this is the particular system of enzyme linked effector system affecting via GPCR mechanism. So friends, as we are talking about the different type of G proteins, so what type of the G proteins? There are certain type of G proteins available, in which the importance are here: GS, GI, G naught, GQ and G13 or G13. These are the major G proteins which are responsible for the different type of cellular function occurring via G protein coupled receptor mechanism. So if we talk about the particular protein, that what type of channel they are opening and what type of work they are doing. So G stimulatory protein, as we discussed, it increase the adenylate cyclase enzyme. AC means adenylate cyclase enzyme, which can increase the production of cyclic AMP. And if the production of cyclic AMP will increase, then that will definitely phospholize the different type of protein via phosphokinase enzyme, and also increase the calcium channels, because G stimulatory protein also increasing the IP3 level. IP3 is the secondary messenger which can release the calcium from the endoplasmic reticulum, as we discussed in the IP3 system. So next, GI, G inhibitory protein decrease the adenylate cyclase, that will opposite effect, that will produce opposite effect. So if the adenylate cyclase stimulation is decreased, so there will be decrease in the level of cyclic AMP, and there should be negative effect. And also increase the potassium channel, and if potassium channel is increasing, opening, and the concentration of potassium increasing, the cell will be again hyperpolarized, not depolarized, hyperpolarized. Means the smooth muscles are and cardiac muscles are going to be relaxed via opening of potassium channel. And again G naught protein and decrease the calcium channel. So it will decrease the calcium channels, and again it will produce the effect like GI inhibitory protein. And GQ is responsible for the increased phospholipase C. If there is an increase in the PLC, so that will again increase the production of IP3 and DAG and also AA, because all three metabolites and secondary messengers are producing via PLC. So if there is an increase in the PLC concentration, so these concentration will be definitely increased. And again G13 will increase the sodium hydrogen exchange. So that will balance the ion, and that will balance the ionic balance. It will control the sodium and hydrogen ion exchange at the cell surface. And beta and gamma, remember, because in the G protein coupled receptor, as we GPCR, they are remain dissociate beta and gamma, and only alpha is movable with the GDP complex. So beta and gamma not dissociate from the cell surface membrane, they will remain associate, but they modulate these channels in mild amount, not majorly. So what are the work of beta and gamma protein? Beta and gamma surface protein act slightly modulate adenylate cyclase, potassium channel and PLC, all three mechanisms, but in the minor manner. So friends, this is all about GPCR. From starting, mechanism of the GPCR, the structure of the GPCR, and the different type of enzyme linked and ion linked effector system which are producing different type of pharmacological and physiological response on the different type of cells and tissue via all these mechanisms. So these are the basically important mechanism: IP3 cyclic AMP production, IP3 DAG system, AA system, and also modulation via ion channels. Sir, thank you.